Hello everyone, my name is Malay Basu. Today I'll be talking about our work on the language of proteins. If you think genomes as a book, then gene or protein sequences can be thought as sentences, and protein domains can be thought of as words. Given an unknown language, for example these plaques from the Indus Valley civilization, the most simplest analysis that can be done is n-gram language modeling. N-gram language model gives the probability of a word given a set of the preceding words. Randomness of a system is measured as entropy. Relative entropy of a language model is the amount of the information gained going from a random arrangement of the words to the meaningful arrangements of the word ordering. This is primarily dictated by grammar. There are around 7,000 languages in the world divided into 19 linguistic families. Now, one very interesting fact about these languages is that all human languages, regardless of the origin, have the relative entropy of 3.6 bits. The reason behind this universal relative entropy of all the human languages is not known, but it has been extensively used in finding out whether a series of symbols represent a language, like the plaques discovered from the Indus Valley civilization. So formally, a bigram language model is the conditional probability of a domain d sub n given its preceding word d sub n minus 1. So we create a domain language model from 450 eukaryotes, 180 archaea, and about 4,000 prokaryotes. So the first question we asked is this: how much information the domain language gained by following the grammar? So when we looked at the relative entropies, we found that except in animals and a small group of archaea, the relative entropy lies in a close range of around 1.2 bits. The phenomenon is very similar to the universal relative entropy as seen in case of natural languages. Now let's see how these grammars evolved. We need to know about cross entropy or perplexity. It's a measure of uncertainty of a genome given a language model from another genome. And by a simple mathematical trick, the cross entropy values can be converted to a distance matrix, and that can be plotted as a phylogenetic tree. So we created the tree for 38 eukaryotic genomes. The trees showed remarkable similarity to existing tree topology. We also used these bigram conditional probabilities to find bigram features or phrases that are most prominent in each major branch of life. The bigram features identified important function in each subgroups, like eukaryotes and prokaryotes are defined by the ubiquitin and spliceosome, the quintessential features of eukaryotes. We therefore conclude that like natural languages, protein grammars do have an universal constant entropy. There exists a quasi-universal grammar of protein domains, and the grammar must have 1.2 bits of complexity to maintain a functional cell. The work described here have been published in these 3 papers. Many people have contributed to the work that I presented. Questions and Rotten Tomatoes.